So now in this video let's see about fruit and seed. So fruit is actually the characteristic features of angiosperms. Angiosperms, we have seen the flowering plants, right? So the formation of fruit is actually a characteristic feature of angiosperms. Actually fruit is a matured and ripened ovary. So ovary will after fertilization. Fertilization is the process of fusion of male and female gamete resulting in the formation of zygote. The zygote will develop into embryo, and this embryo will develop into new plant. Actually embryo will be in the seed, that we will see in the seed part. So what you have to understand here is the fruit is a ripened or matured ovary, and it is formed after fertilization. And there are some cases where the fruits are formed without fertilization. In that case, that kind of fruit is known as parthenocarpic fruit. So if the fruits are formed without fertilization, it is known as parthenocarpic fruit. If you take a fruit, there will be a wall, and in the center or anywhere you can see a seed. So a fruit usually consists of a wall and seed. This wall has a special name called pericarp. So the wall of the fruit is known as pericarp. And you can see three layers. So first one being the epicarp, which is the outer layer, and middle layer is mesocarp, and the inner layer is endocarp. A fruit generally consists of a wall, which is known as pericarp, and a seed. The wall, it has three layers: the outer epicarp, middle mesocarp, and inner endocarp. There are see here two examples are given. One is mango, another is coconut. So this two are commonly known as, it belongs to a type of fruit called drupe. So if you see, it will have monocarpellary superior ovary. The ovary will be monocarpellary, and the ovaries will be superior. That is, ovary will be on the top, all the other parts will be at the bottom. Here the epicarp, that pericarp is well differentiated into epicarp, mesocarp, and endocarp. You can clearly see this in mango. So outer layer will be the greenish or reddish epicarp, and the middle fleshy part which we eat is actually mesocarp. And over the seed you can see a layer that is somewhat papery or hard, that is known as endocarp. So in these two cases you can see the three layers clearly, right? So in the case of this coconut, the middle fleshy mesocarp will be transformed into this fibrous part. So this is actually the mesocarp, and in the mango you can see a hard stony seed also. Yes, so in these two cases you can differentiate clearly the three layers of pericarp, and they are denoted by a special term drupe. So they are monocarpellary. So they are monocarpellary and superior ovary. So they develop from these kind of fruits, they usually develop from monocarpellary superior ovary. So what is seed? So this is the main part of a flower, and the purpose of flower is to produce seeds, because seeds are the only agents that will produce the next generation. So what about seed coat? It has two important parts. One is seed coat, that is the outermost layer, and an embryo, which is the young plant which will develop into the mature one. So if you take an embryo also it holds three layers. So first one being the axis. So this is the embryonal axis. It is said to be embryonal axis. On the top portion will develop into plumule, and the bottom portion will develop into radicle. The plumule will give rise to shoot system, and the radicle will give rise to root system. So there are two kinds of seeds. So in a seed there is a structure called cotyledon, which will store food materials. So whenever needed, the embryo will use this food material for its growth. So if there are two cotyledons, that kind of plant is known as dicotyledonous seeds. So if the seed has two cotyledons, it is known as dicotyledonous seed. And if the seed is having a single cotyledon, it is known as monocotyledonous seeds. The seed coat is again divided into, so if you take a seed coat, it is again divided into two layers. The one is testa and the inner tegment, testa being the outer layer and tegment being the inner layer. So you can see the seed is attached to some part in the fruit, right? So it will leave a scar in that part where it is getting attached to the fruit. So you can see clearly in a bean, here there will be a white scar. So that part is actually known as hilum. So this indicates it is attached to the placenta. You can clearly see that, right? So placenta is actually present in the ovary. Ovary is what developing to fruit. So obviously it is a part of a fruit. So in the case of bean, you can see a white structure here. So that is actually a scar formed in the place where the seed is attached to the fruit. And above the hilum, so if you see here, so if this is hilum, there will be a small opening called micropyle. And I have already said the embryo will have three things. So one is axis, another is plumule, another is radicle. Apart from this, in some cases like castor, so previously while we are seeing the characteristics of plants, we have discussed the structure of an egg. So in the center there will be two nuclei called polar nuclei, which will fuse together to form a secondary nucleus. So here after fusion of this polar nuclei, the secondary nucleus will be there. So actually this is our main part, egg, which will fuse with the male gamete to develop into seed, and these are vestigial organs. Antipodals and synergids are vestigial organs. So when the pollen is released, it will release two male gametes. Okay, so one of the male gamete will fuse with the egg, giving rise to seed, whereas the other male gamete will fuse with the secondary nucleus and give rise to endosperm. It is actually an additional nutritionous tissue that provides nutrition to the developing embryo. So the two male gametes will be released, of which one will fuse with the egg and the other will fuse with the secondary nucleus. The one fusing with the egg will develop into the seed. The other, which is fusing with the secondary nucleus, will develop into endosperm. Endosperm is actually it has stored food materials which is useful for the growth of the seeds. The plants which is possessing this endosperm are known as endospermic seeds, and the seeds in which the endosperm are absent are known as non-endospermic seeds or non-endospermous. So structure of monocotyledon seed. So it has only one cotyledon. And the seed coat, if you take a maize, rice, you can see that outer papery cover. So that is actually a seed coat. It is membranous, right? And it is usually fused with the fruit wall. And here the center endosperm will be bulky and it stores food. So actually this endosperm is covered by outer protein layer known as alluron layer. So this outer layer is known as alluron layer. Actually it is a protein layer. It produces enzymes that actually breaks down the stored food in the endosperm, which is used by this developing embryo. So here in a small portion the embryo is present, but here there are two special things. As usual, here there is a plumule which will develop into shoot system, and this is radicle which will develop into root system. But each of them have a separate covering. So you can see something covering this, right? So the part which is covering the plumule is actually coleoptile, and the part which is covering the radicle is coleorhiza. So this coleoptile and coleorhiza, the main function of both is to give protection to the delicate tissue which is growing inside. So this coleoptile, when the seed is sown in the soil, it has to come out of the soil, right? So that time it will penetrate and come out of the soil, by at the same time it will protect the delicate tissue inside. Same way this radicle, it has coleorhiza. First thing, it protects the radicle and its delicate tissue, and the other, it will penetrate the soil, and the main important part of function of this is it will fix the seed to the soil. So it will help in anchorage.